Good morning students, my name is Kifayat Hussain. I have done MA Sociology, MSc Biotechnology and have also qualified CSIR NET. I am today going to deliver lecture on DNA replication, which is a very complex process. DNA replication is involved in growth as well as cancer. So it's a very important topic. Most of the questions, if you don't understand DNA replication properly, so these questions will come in the competitive exams and you will be unable to answer such questions. First of all, we should know the basics how DNA replication takes place. Certain things should be kept in mind before we begin initiation, elongation and termination of DNA. First of all, DNA replication is semi-conservative. DNA replication is semi-conservative. That means one of the parental strand acts as a template to synthesize the new daughter strand. This is known as semi-conservative. Second is semi-discontinuous nature of DNA replication. DNA replication is not always continuous process. It is a discontinuous process. It was observed. One of the problem which is faced by DNA polymerase, which I will explain later, is DNA polymerase always extends or elongates the DNA chain, DNA strand in 5 to 3 direction. Because the two chains are antiparallel, I will show you what does antiparallel means. One chain runs into the 5 to 3 direction and other runs into the 3 to 5 direction. This is known as antiparallel nature of the DNA. So one of the strands, DNA polymerase always polymerizes the reaction in 5 to 3 direction. So one of the strands which is continuously synthesized is known as leading strand, and other is known as, which is synthesized in fragments, is known as lagging strand. So it is a semi-discontinuous process. We should be knowing each and every enzyme which is required in DNA replication. Let us go to the board. These are the enzymes which are required for DNA replication. Helicase is the enzyme which unwinds the two strands so that strands act as a template to synthesize the daughter strands. It creates a replication fork. So helicase is responsible for unwinding DNA and creation of replication fork. Primase. It should be noted that DNA replication cannot take place without the help of a few 10 to 12 RNA strand, which is known as primer. Here I have shown it in a red color. So this primase synthesizes this 10 to 12 nucleotide RNA, which is required both once by leading strand and every Okazaki fragment when it is synthesized, this primer is required. So primase is required for DNA replication as well. Together these helicase and primase are known as primosomes. Now the most important enzyme, that is DNA polymerase. There are three types of DNA polymerase: DNA polymerase first, DNA polymerase second, DNA polymerase third, which we are discussing about E. coli. E. coli. So DNA polymerase has got three functions. One is polymerization of the DNA. For example, I have taken this strand: adenine, guanine, adenine, adenine, guanine. Suppose these cytosine and thymine are not present there. These are added by DNA polymerase and the reaction continues so that the strand is extended. Second important, most important function of the DNA polymerase is 3 to 5 proofreading, which is most important. Proofreading means if a wrong base is inserted, it will create a mutation. That mutation may lead into the further in humans or in any other mammal, result into the cancer formation. So these bases, this base is removed by DNA polymerase. It has got 3 to 5 exonuclease activity. 3 to 5, not 5 to 3, 3 to 5 exonuclease activity. Here I have shown a strand: adenine, thymine, guanine, cytosine. Here instead of cytosine, uracil has been added, and due to proofreading activity of DNA polymerase will come from 3 to 5 side and remove this uracil so that the proofreading is done. The accuracy of DNA is 10 raised to power minus 5. If in one run from 5 to 3, when DNA replication takes place, 10 minus 5, its capability to insert a wrong base is 10 minus 5. From 5 to 3, it has got also 10 minus 5. So total 10 into 10 is equal to 10 raised to power 10. So in 10 raised to power 10, one nucleotide, wrong nucleotide, may be inserted. Now another enzyme, DNA gyrase. DNA gyrase, what it does? DNA gyrase relaxes the supercoils. Once the helicase begins the job of forming replication fork, replication fork is formed here and here. If you consider it a rubber band, so there will be strain in the DNA and supercoils will be formed. For example, you take in your hand a rubber band and try to unwind it 10 or 20 times. Then try insert your hand into the rubber band and try to unwind it. There will be supercoils formed. So these supercoils are relaxed by DNA gyrase. DNA gyrase, if certain in medicine, certain antibiotics, for example norfloxacin, acts on this DNA gyrase so that in bacteria DNA synthesis does not take place and they die and infection is cured. Now last but not the least, DNA topoisomerase fourth. When the DNA replication fork reaches the site of third site where the replication ends, these become entangled. DNA strands, circular strands in E. coli become entangled. These are entanglement are these are separated by DNA polymerase fourth. DNA replication is a very complex process, but for the sake of convenience, it has been divided into three processes. First is initiation, second is elongation, third is termination. Let us first take the initiation process. Initiation always begins at ORC, or origin of replication. Origin of replication, or ORIC, where there are sites, in both eukaryotes and prokaryotes, where origin of replication lies, where from the replication bubble extends in the both ends and DNA replication proceeds. We are discussing here E. coli because it's a simple model. So here in initiation, the enzymes required are HU, WHO, DNA, helicase and primase. WHO protein, what it does, it bends it. I have taken this, you can see this ORC as here as a fragment. WHO protein bends the DNA, here you see, so that it destabilizes the helix. Then DNA comes and melts 13 to 12 base pairs left to the origin of replication. This is the initiation of DNA replication. Then later DNA attracts helicase and primase, which begins the initiation of DNA replication. Now I will talk about second process of DNA replication, that is elongation. Elongation simply means extension of the DNA of the primer to primer. New nucleotides are added so that the base second template is created. This is elongation, called elongation of the DNA. So how elongation takes place? As I already told you, chromosomes, helicase and primase begins the initiation. When helicase unwinds the DNA, it creates the replication fork. So primase, the enzyme which is required for the synthesis of primer, otherwise DNA replication would not take place. DNA polymerase always requires a primer, so that a free end, so that to that end DNA nucleotides may be added. So this helicase, when this unwinds the DNA, it creates a tension in the DNA, which results in the formation of supercoils. These supercoils are relaxed by DNA gyrase. One thing should be remembered, as I already told you, that DNA replication is not a continuous process, it is a semi-continuous process. DNA polymerase enzyme, which I already mentioned, always extends the DNA 5 to 3 direction. So one of the template provides the 5 to provides a free 3 end. To that end nucleotides are added continuously, so this strand is synthesized continuously. This strand is known as leading strand. Other strand which is synthesized discontinuously is known as lagging strand. In this strand, 1000 to 2000 nucleotides are added each time by DNA polymerase. However, each time a DNA primer is required, which is synthesized by DNA primase, this primase enzyme. These fragments of 1000 to 2000 length are known as Okazaki fragments. So these fragments are known as Okazaki fragments. Now the speed. What is the speed of DNA polymerase? DNA polymerase almost in vitro polymerizes almost 1,000 nucleotides per second. Now, due to 3 to 5 exonuclease proofreading activity of DNA polymerase, it removes these primers from these Okazaki fragments, and DNA ligase, an enzyme which creates phosphodiester bond between the two DNA Okazaki fragments. So it results into a continuous DNA template. Now let us discuss the last process of DNA replication, that's known as termination. DNA replication in E. coli, the DNA is circular in shape. So when the replication fork meets at a point where sites are known as third sites, where termination sites also, the DNA gets entangled. So they are not separated on their own. An enzyme is required that is known as DNA gyrase 4th, which separates the two DNA strands, creates a parent and daughter strands. DNA replication in eukaryotes is a very complex process. However, the process we studied in E. coli, same initiation, elongation and termination occurs in DNA replication of eukaryotes. In prokaryotes like E. coli, there is only one origin of replication, only one replication bubble. In case of eukaryotes, there may be many replication forks formed in a single chromosome. There are three types of enzymes, only three types of enzymes in prokaryotes: DNA polymerase first, which require DNA polymerase second, and DNA polymerase third. DNA polymerase second is the real polymerase which carries out polymerization of the DNA. However, in case of eukaryotic DNA replication, there are many types of DNA polymerases. First of all, we will study DNA polymerase alpha. Its job is to synthesize primers, priming of DNA. DNA polymerase delta. Its job is elongation of lagging strand. DNA polymerase epsilon. Its job is elongation of leading strand. DNA polymerase beta. Its job is DNA repair. DNA polymerase gamma. Its job is replication of mitochondrial DNA. In eukaryotic replication, one more thing which has to be kept in mind, that ends are not synthesized by DNA polymerase. So that means with each replication, the DNA will get shorter and shorter and shorter and shorter. So how this problem is solved? There is an enzyme known as telomerase. Telomerase has got its own RNA template which guides it to add this missing nucleotides of telomeres. Ends of the nucleus, ends of the chromosome are known as telomeres. So these are the telomeres. This brings the end of the DNA replication. I hope you understood what I said, and I hope in near future lectures I will deliver some more lectures on on biology, inshallah, very soon.